Chapter 8 in the text goes into some lovely, gritty detail about the development of the nervous system, and we are just going to briefly discuss it and talk about it a little bit in the different phases, but let's look at the learning objectives so we know how to focus our studies. So I want you to be able to describe how neuron death and axon retraction sculpt the developing nervous system. In the module, there is a cool 1-minute video of the development of a neural growth cone. It's pretty cool. Take a look at it if you get a chance. I want you to know at what point in the developmental process the axons begin to be myelinated, and how long does it take the process of myelination to complete, and during which period of the development is the central nervous system susceptible to major malformations versus minor malformations. So we'll talk about which week of development that happens in. So humans in utero undergo 3 developmental stages. There's the pre-embryonic stage, which happens between conception and up to day 14. Embryonic, which is day 15 to the end of week 8. And the fetal stage, which is week 8 to birth. Okay. The embryonic stage is from day 15 to the end of the 8th week. That's the stage where the organs are starting to be formed. In the fetal stage, from the end of week 8 until birth, the nervous system develops more fully, and that's when myelination begins in the fetal stage. And it actually keeps going until after birth. So formation of the nervous system occurs during the embryonic stage, and it involves 2 phases: the neural tube formation, which is days 18 to 26, pretty early on, right? And brain formation begins on day 28 and continues during the fetal stage. So lots of super important stuff going on at the beginning of the embryonic stage. So all vertebrates look like this at some stage in their development. As the neural tube closes, the adjacent mesoderm divides into somites, which become bones, muscles, and dermis. So the nervous system and the bones, muscles, and skin are intimately related from the very beginning. As the cells of the inner layer of the neural tube proliferate, grooves form on each side, and those separate the tube into ventral and dorsal sections. And the ventral and dorsal sections eventually become the motor and sensory systems. The neural crest separates into 2 columns, and peripheral sensory neurons convey information from sensory receptors to what they call the association plate. Do I want you to know all this detail? You do not have to know all this detail. It's not in learning objectives, right? So you don't need to know it. But just to give you an idea of there's a lot of complex stuff going on, organizing things so early that you can't tell if this is another vertebrate or a human being, because we all look like this at some stage. Pretty interesting, right? But at that point, we have brain formation. So the hindbrain divides into 2 sections, and the structures differentiate later to become the medulla, the pons, and the cerebellum, which is a pretty important part of the brain. Brain development continues during the fetal stage, and hopefully brain development continues throughout our life. So let's work on that, shall we? At the cellular level, there's a lot going on too. So the neurons have to differentiate appropriately after migrating to their final location. So the function of each neuron depends on the area of the brain where the neuron migrates. So at first they're just all undifferentiated, and then they migrate to their final destination, and then they develop their function. So there's a process that emerges from the neuron that is called the growth cone, and that's that video that's in the Canvas module that's pretty cool. So the growth cone, it's out there, it's seeking chemical messengers, it's trying to find its target cell. When it contacts its target cell, it starts to form synaptic vesicles, and it starts to become the presynaptic membrane, which is pretty cool. So we have these new neural connections going on. So a lot of the new neural connections die. Up to half in some areas. And the connections that fail to form optimal connections or are too inactive just die off. So we start out with lots more connections than we need, and we sort of trim it down to what we do need. So neuronal death and axon retraction sculpt the developing nervous system. When I said in a previous lecture that neurons that fire together wire together, if the more those neurons are used, the more their chance of being kept is. So some neurons retract from some areas but maintain contact in other areas. During development, there might be several axons that innervate one muscle cell, but by week 25, there's only one axon per muscle fiber, as is in the mature nervous system. And whether that muscle fiber becomes a type 1 or a type 2, a fast twitch or a slow twitch, depends on what axon innervates it. So we start out with a lot of connections, and then we trim it down by neuronal death and axon retraction into what we really need. So the neuronal connections sculpt the developing musculature. The muscle fiber type is dependent on innervation. So a fast twitch muscle can be converted to a slow twitch muscle if it's innervated by a slow motor neuron. And a slow twitch muscle can be converted to a fast twitch muscle if it's innervated by a fast motor neuron. So it starts out with a lot of connections, 25, and depending on which ones are used, that's how it develops. So it's pretty interesting, I think.